and welcome to the ACR Journals on Air podcast. I'm your host, Vicky Shamagam. The beautiful thing about science is that the data is always telling you something. It might not be telling you the answer to the question you had in mind when you designed your experiment, but it's always telling you something. If the experiment was well executed, we just have to open our minds, let go of our narratives, and consider that the data may be an important finding in a different scientific puzzle than the one we were originally thinking of. This is what struck me when I read a manuscript that was recently published in ACR Open Rheumatology. The manuscript is entitled A Human MSH6 Germline Variant Associated with Systemic Lupus Erythematosus Induces Lupus-like Disease in Mice. And the senior author is Dr. Joanne Sweezy, director of the Cancer Center and the Nancy C. and Craig M. Burge Endowed Chair of Cancer Biology at the University of Arizona Medical Center. And it's a pleasure to have her on the show today to talk about this paper. Welcome to the show, Joanne.
Thank you. It's great to be here.
So, Joanne, you are a cancer biologist, and MSH6 is a gene known to be important in the repair of genetic mismatch. It's actually one of the group of genes implicated in the familial cancer syndrome known as Lynch syndrome. For rheumatology listeners who may be rusty on this, Lynch syndrome is the most common cause of inherited colorectal cancer and should be suspected in individuals with colorectal cancer prior to age 50. And there are a number of other Lynch-associated cancers, including endometrial, ovarian, stomach, small intestine, renal pelvis, or ureter. And studies have shown an association between Lynch syndrome and germline mutations in DNA mismatch repair genes, of which MSH6 is one. So, Joanne, to get us started, can you help our listeners understand the initial genomic analysis that your team did on patients with lupus, and how did that collaboration come about?
Sure. So the way the collaboration came about was really one of serendipity. That's how I would describe it. As you mentioned, I'm a cancer biologist, and for years we've been studying germline and somatic variants in DNA repair genes that are associated with cancer, in other words, found in tumors, or that could potentially play a role in cancer development. And one of these was in a gene called PALB that encodes for a DNA polymerase called DNA polymerase beta, which is very important for DNA repair. We'd been working with that variant for quite a while in cells and showed that it caused genomic instability and it induced increased proliferation and other hallmarks of cellular transformation. In other words, a more cancerous phenotype. And so we decided to really ask the question, does this potential germline variant in the PALB gene induce cancer in mice? And so we made the mouse model, and we waited and waited and waited. Mice never develop cancer. As a matter of fact, we keep them running over 10 years. They've never developed cancer, but they develop lupus-like disease, an autoimmune syndrome, which we were quite surprised about. And these mice have skin lesions, they have high levels of autoantibodies, and they have lupus nephritis, and also aberrant VDJ recombination and somatic hypermutation, an aberrant immune repertoire. So we were in touch with colleagues at Yale University and who said to us, you've got to meet Dr. Lindsay Criswell. She is a person you need to collaborate in terms of the genetics of DNA repair in lupus. That's really how we got into this whole field of lupus. We started out in cancer and now we've moved to autoimmune disease.
And so that was a different set of mice than the ones that you're describing in this experiment, right?
Completely different set of mice.
So that's interesting. And so in this experiment, you lined up an initial genomic analysis in patients with lupus and then worked backwards to study the genes that you found in that analysis. I think you looked at somewhere in the region of 114 SNPs in 10 different mismatch repair genes and identified some variants that were enriched in the lupus subjects compared to controls. Can you tell us a little bit about those and then what prompted you to move forward with the specific MSH6 variant?
So we initially proposed to Dr. Criswell and her team, including Kim Taylor, that we should potentially we couldn't look at all the DNA repair genes, over 300 of them. And so we decided to look at DNA repair genes that also had a role in the development of the immune repertoire. And among those genes are MSH2 and MSH6. They play a role in somatic hypermutation and class switch recombination. So that's why we selected those genes and others. There are also others in that study. We then found this MSH6 S5063 variant, as you mentioned, and we were very interested in this variant because it has a significant odds ratio, even though there are very few numbers of individuals who actually have the variant. But what really struck us was that the carriers of this variant were significantly younger than the general lupus cohort. So there are actually probably teenagers in this cohort of individuals with this particular variant. And we thought that was a very interesting characteristic. And so we decided to move on with studies of this particular MSH6 variant.
And that's interesting. I have a couple kind of follow-up questions about those patients who happen to be carriers of this variant. Did you see higher rates of lupus nephritis or autoantibody production in those patients who carried the variant?
Compared to controls, but not compared to individuals with lupus.
So not compared to the larger population of patients that you were studying?
Correct.
To be sure, those people who carried those mutations, did they have any kind of family history of the Lynch syndrome cancers or malignancy, or do you just not have that data in the lupus cohort?
We don't have that data in the lupus cohort, but what I can tell you about this particular gene, I researched this MSH6 variant very widely, actually, and it turned out that a lot of biochemical assays had been done with this variant that showed that it had no effect on mismatch repair. And I was actually encouraged by folks in the mismatch repair field to not work on this variant.
So isn't that sometimes where you find the interesting science is when somebody says, no, don't study that, it's a waste of your time. And
yes.
You're like, no, there's something to be found.
Yes.
So that's really interesting. So the association really between the MSH6 variant and the early onset of lupus in the patient cohort is what prompted you to move forward with your new mouse model to see if you could establish using CRISPR-Cas9 technology and giving the mice this missense mutation, whether you could create the lupus-like phenotype in your mice. Can you tell us a little bit about that process and then how you evaluated the progeny after you generated the mutation to confirm you had the expected genotypes and protein expression?
Sure. So right now it's actually pretty straightforward to generate mouse models using CRISPR-Cas9. Most institutions, including the one I'm at now, have core facilities that do it. And you can usually get the mouse in about 6 weeks, which is fantastic. So we had the mice pretty quickly and bred them up and monitored autoantibodies and saw that they were developing increased levels of autoantibodies. Not all the mice. So these are very heterogeneous, sort of like human beings. We don't know what flares are in mice, at least to the best of my knowledge, but they started to develop it. I think the surprise came when none of these mice exhibited lupus nephritis, as typical mouse models of lupus do. These mice didn't. And we made the observation that the MSH6 protein is not highly expressed in the kidneys of mice, nor is it highly expressed in the kidneys of humans. And so to us, it suggests that this variant would have to be expressed in the kidney in order for the mouse to develop a phenotype.
But you did find both presence of autoantibodies and then that these mice had some lung issues, correct? Can you tell us what you saw in the lungs of these mice and how you interpreted that finding?
The lungs are very interesting. So the mice die a little earlier than their control litter mates. And the lungs of these animals have an immune infiltrate that is composed of monocytes. We now actually think that these are aberrant alveolar macrophages. In data that's unpublished at this point, we've started to look at gene expression and we've started to look at cytokine profiles, and it's early days, and so I really don't have an interpretation for this. We really think that there is an aberrant immune response in the lungs, and we think it's probably a response to, even though these mice are living in essentially a very protected living situation, there are probably things in their environment that they're breathing in. And so the immune system is constantly responding to this, but it responds and then it's dampened down, and it responds and then it's dampened down. And we think there's something wrong with either one or both, the pro-inflammatory or the anti-inflammatory response. And in the paper, we show that we can get there are various types of macrophages in these lungs, pro-inflammatory and anti-inflammatory. So we're in the early phases of this. But interestingly, we've been told that individuals with lupus actually can have increased lung disease and inflammatory lung disease.
So I think at this point, it's going to be quite helpful to bring in some rheumatologic perspective to this discussion. And I'm delighted that Dr. Wesley Reeves, Section Chief in Rheumatology at the University of Florida, is able to join us. Now, West, you may not know, but my maiden name is Reeves. So probably somewhere way back, we share some genetic mutations, but hopefully not in genes associated with mismatch repair. Thank you so much for joining us today and welcome to the show.
Thank you very much.
So I'm interested to hear your thoughts on this manuscript, and I'm wondering if you could first tell us how you interpreted the human exome chip data that was presented and what that could mean in the context of managing lupus patients. Are these mutations things we should be looking for, and particularly in the pediatric and adolescent population presenting with lupus at a young age? If so, how should we be counseling those patients and their families in regards to long-term implications of these mutations?
What was striking to me was the extremely high relative risk of this variant with lupus-like disease. It's much higher than what you see for standard genes that are identified by GWAS. So it's a very strong factor. And it was curious that it hadn't been identified previously in the GWAS studies. But there's a lot of evidence that this particular gene may be doing something. And so there are actually two syndromes with MSH6 mutations. The first is the Lynch syndrome, which you talked about, which is caused by a heterozygous germline mutation. And it's associated with a really high risk. I mean, it's 70% over your lifetime of developing colorectal cancer, usually in the fourth or fifth decade. Also, increased risk of endometrial cancer. And the tumors arise when there's a somatic mutation of the second copy of the gene. So the tumors will have a germline mutation and then a somatic mutation in general. So this is really the first study that I'm aware of that links heterozygous germline mutation of this gene with lupus. Now, there's a second disorder which is caused by biallelic germline mutations in these mutations, and this is called constitutional mismatch repair deficiency, or CMMRD. It's a rare disease. It's associated with consanguinity, and it's associated with very early and broad spectrum malignancy, more broad than what you see in Lynch syndrome, and they get a neurofibromatosis-like disorder with café au lait spots. The most common malignancy is a brain tumor, actually, in this syndrome. And if you look at the literature, although it's really rare, 2.5% of patients with this biallelic disorder have pediatric lupus. It's really a high percentage. Interestingly, the mutations that these kids have are different than the ones that are reported in the current paper. But 5 out of the 6 patients that have been reported with this disease, along with pediatric lupus, have mutations in MSH6. So there seems to be a predilection for, although there are a bunch of genes that cause this syndrome, really MSH6 is overrepresented. So genetic counseling is complicated. I think it's been, so I'm not a geneticist, but it's been proposed that the genetic counseling should be offered first to the parents of the patients because they're going to be carriers of a potential cancer predisposing gene. The heterozygous parents probably should be monitored for colorectal and endometrial cancer, although it's too early to tell whether this particular mutation is associated with that. The role of genetic screening with this particular variant is less clear than it is with some of the other variants. But at least the mice didn't have increased risk of getting gross tumors. That doesn't mean that they don't have microscopic tumors hiding someplace. So I think the whole issue of genetic counseling in this situation is a bit up in the air, but I think it probably would be worth considering genetically counseling the parents of these children.
And actually, since you bring up the mice here, I'm kind of interested by your perspective on the mice that were generated in this experiment. There were a lot of them, and they were obviously meticulously genotyped and phenotyped. And I guess my first question is, I know you're not a mouse rheumatologist, but as a translational rheumatologist, do you think the mice had lupus or something else?
I think that's an open question. So I think a lot more needs to be done. The mice don't meet criteria that have been established for human lupus. I mean, the first criterion is a 1 to 80 ANA, and these mice were screened at 1 to 50, so you don't even have the entry criterion here. In my opinion, this may be an example of what's been termed monogenic lupus. There are a bunch of syndromes where you have defects in a single gene that cause something that reproduces some of the clinical features of SLE. This could be an example of that. The ANA data, I think, are important, but are fairly preliminary. So there are a number of things that you would want to know. First, you want to know what the ANA titers are. So how high is this, and how does it compare with the kinds of titers that you see in other mouse lupus strains, so NZBW or MRL-LPR. You'd want to know what patterns these ANAs give in indirect immunofluorescence, and particularly importantly, you'd want to know what the fine specificities are. Are they the typical lupus autoantibodies like anti-DNA and anti-SM, or are they against random antigens? So the random antigens, on the one hand, it doesn't sound as interesting, but on the other hand, it may tell you something about the pathogenesis, because presumably these somatic mutations, if they occur with this variant, can occur in pretty much any cell. And there are a number of papers that suggest that if you get a somatic mutation in a self protein, now this self protein is no longer strictly speaking self, and it could potentially target the protein for autoimmunity by making the proteins more immunogenic. And then the final thing I think that you would like to know is, are these antibodies associated with interferon signature, which is really important for the association of things like anti-DNA and anti-SM with lupus. So the interferon signature positive subset of patients has a much higher prevalence of those antibodies. And so that would be useful. The same problem exists with the lung disease. So right now, I don't see a strict correlation with the usual types of lupus lung disease. So there was no diffuse alveolar hemorrhage in these mice. The most common manifestation would be pleuritis, but they didn't look at that, or interstitial lung disease. There are features of both M1 and M2 macrophages. These are the pro and anti-inflammatory flavors of macrophages. But the macrophages were located inside the alveoli. And if you look at the figures, these macrophages are huge, and they're inside the alveoli. And it suggests, as previously alluded to, that these may be alveolar macrophages, which are also CD68 positive. So increased numbers of alveolar macrophages are uncommon in lupus, and it would suggest that there's something going on inside the alveoli. So is there an infection there? Are the lung epithelial cells, which is on the side of the same side as the alveolar space, are they mutating? Are they becoming inflammatory? And are these macrophages trying to clean up the damage to mutated alveolar epithelial cells or something? I think there's a lot that still needs to be determined about both the immunological disorder and the lung disease. It's an intriguing paper that may reproduce some of the manifestations that you see in lupus.
Yeah, and thank you for that incredibly detailed response. I agree. I think the data are really interesting, right? And we do have a couple of diseases where lung inflammation is the really prominent organ inflamed in autoimmune disease, and some of those that are associated with contemporaneous malignancy development, of course, myositis in that group and scleroderma. And I'm wondering if these mismatch repair genes have been studied at all in those two diseases, and whether these particular mice might well be a better model for those diseases than for lupus.
Yeah, so I did a literature search on that and didn't find anything.
Yeah.
I think that also is an open question. I would add Sjogren's syndrome to the list of diseases with malignancy and lung disease, but what you see in Sjogren's syndrome is a lymphocytic interstitial pneumonitis. It's not particularly characterized by macrophages, so it doesn't really resemble what you see in Sjogren's. Myositis also is associated with lymphocytic attack in the interstitium. So it's not inside the alveoli, it's in the interstitium of the lung, and you get an interstitial lung disease. Same thing with scleroderma. So the alveolar pattern is interesting. It's a little unusual in autoimmunity. And then I also did a search to see what I could find about mismatch repair and scleroderma and polymyositis, and there just isn't anything. It's a wide open area, yeah.
No, I looked the same and wondered also. And Joanne, I'm going to bring you back into the discussion because as I read about your mice, and I clearly think you've found something and something really interesting, I wondered, did you go back at all to the patients and try and delve down any deeper into defining whether they grew up with a manifestation that truly looked like lupus, or did they morph into something that was more of an overlap syndrome? We in rheumatology are pretty comfortable with these people that start off looking like one autoimmune disease and kind of morph into more of a mixed picture. Do you have any information about those kids as they grew up?
Not yet. That's what we're doing now. We're really trying to find more information about these folks to see if the mouse model is recapitulating the human disease at all. May I go back and talk about the types of antigens?
Absolutely.
We didn't publish this, but in terms of the types of antigens, we see increases in everything. So it's not anything specific. I think that would suggest that there could be a mutagenic basis to this, but we've done everything we can to look at mutagenesis. We just don't see it in these animals. And actually, that's something that I've been trained in. So we don't see increased mutation frequencies of mutagenesis of any type, because that's exactly what we thought was going on. And we're perplexed by this. The biochemical assays also suggest that this protein is working normally as a mismatch repair protein. So the biochemistry agrees with what we're seeing in the animal. So it's very curious to us.
Yeah.
I think that's fascinating. And I think this work was obviously years of work, multiple collaborations at many institutions, and has obviously perhaps not quite yielded the results you were anticipating when you designed the experiment, but I'm sure it took an enormous amount of effort to get this through to publication, and perseverance is so important for translational research. I'm wondering, Joanne, would you be willing to share any tips for other researchers on how to see things through, even when data is showing you something that's not quite what you expected?
Sure. I've learned in my career that that's the most exciting part of what I do, is when I test a hypothesis, and instead of rendering it null, I'm going in a completely different direction. And I think what I've learned from this is to reach out for help to experts in the field you think you have now wound up in. I think it's just so important to bring that perspective to the table. And it's been so helpful to work with Dr. Criswell and her colleagues on this. It's been a pleasure to work with them and to learn more about this. And now we're making more mouse models. It's a very interesting field.
It is. And there are actually a number of rheumatologists now studying malignancy-associated autoimmunity. So I'm hoping that perhaps your cross-pollination of us by coming on this podcast might actually generate you a couple new collaborations as well. And West, you conduct both translational research, review translational manuscripts and research grants. Do you have any tips for investigators on navigating unexpected results?
I think the key to the whole thing from a non-geneticist perspective is really detailed clinical and immunological phenotyping. There may be a very small subset of lupus patients that look exactly like what you're seeing in a mouse model. It's unlikely that any one mouse model is going to explain everything in lupus. And so you can use the mouse models to tell you something about the subset that they resemble, but that involves really detailed phenotyping of what's going on in the mice. It should also be kept in mind that the mutation of the same gene can cause different syndromes in humans and mice. So a good example of that is MRL-LPR mice. So the MRL-LPR mice have a mutation in FAS. They get rip-roaring lupus. So they have really bad glomerulonephritis. They make anti-SM. They make anti-double-stranded DNA antibodies. They really do mimic the autoantibodies in the renal disease. But there are humans with FAS mutations too, and most of the humans with these mutations get hematological autoimmunity. It's really unusual for them to get card-carrying lupus from their FAS mutations. I think the animal models can really help you to understand in more depth what's going on in these subsets of lupus patients, because lupus is ultimately a really heterogeneous disease. And they raise the potential that there may be modifying genes, or that there could be pathways that duplicate the pathway that is mutated in one species that maybe are not active in another species. So I think the animal models can really contribute to understanding the complexity and heterogeneity of lupus, but you have to spend the time to look very carefully at what the phenotype is, what the real phenotype in the lupus patients is as well, and try and make a match between a subset of lupus and the animal model. So that's what I advise people in the lab, who are working. We do a lot of work on animal models, and that's what I advise them to do, is to scrutinize the clinical phenotypes in the mice.
I really think this has been an incredibly interesting discussion. I want to thank both West and Joanne for joining us today on the podcast. I do want to close this episode by thanking people who are brave enough to do cross-disciplinary research, advancing the science sometimes in unpredictable ways. And I think it's incredibly important to recognize that in experiments and in life, sometimes results are not what you expect, and that time and space allow us to interpret our results through a different frame of reference. In that regard, science reminds me a little bit of sailing. Sometimes we have to let the wind carry us in a different direction so that we can ultimately get across the ocean. Thank you all for listening to this show. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at ACR Room on Facebook and Twitter, and please rate and subscribe to our podcasts at acronair.org or wherever you get your podcasts.